All right, if you're all set, send your note cards to the center.
That's right there.
It just has to be interesting to me. It could be like, I broke my ankle one time. It could be the weirdest I've ever pretty close to the All right, was everyone able to successfully find my syllabus on the faculty web for those who are looking for it? Okay, you don't need to have a hard copy of it, you just need to know where it is. I'm going to point out some of the most important things on the syllabus. I'm Dr. Srovic, in case you guys haven't had me before, so I'll be teaching the class, which is why my name's right there. This is an introductory course to the field of biochemistry. How many people are going to take biochemistry lab? The lab. 4 or 5? How many people are going to take the second half of the lecture course? About the same people. Okay, cool. So this course covers everything about introductory molecules, lipids, fats, proteins, membranes, a smidge bit of DNA, RNA, and then we cover citric acid cycle, oxidative phosphorylation, and a few other cycles like that. So we move pretty quickly because we're going to do 12 chapters this semester, which is about a chapter a week. So speedy. It's a speedy course, which if anyone has a friend in medical school, ask them about their biochem course. They'll tell them that the 12 chapters we cover in 14 weeks, they cover in 1 week, and they consider it review in medical school. So they're going to walk in and say, you guys have already seen this, here's your test. We'll talk about something else next Monday. It's similar in vet school. This is just going to be review when you get there. So while this seems fast, it only gets faster when you get to vet and med school. I don't know about dental school. I don't have any friends in dental school. So, all right, the first page is all the same standard stuff. We're going to assess you using quizzes, lab reports, if you're taking the laboratory portion of the course, group work, exams. You need to have a C or higher to officially really get credit for this course. Let's see, on the next page, we are using this textbook. If you have the second edition, that's fine, as long as you recognize that the homework questions I'm going to be assigning may or may not be the same. So just make sure you have a friend somewhere in one of the 3 sections that has a copy of the third edition, and you can. I've had students take camera photos of my homework page. Like, if you don't have this edition, you can come to my office, use your phone, take a picture of the questions, or get someone else who has the textbook and use their version. Whatever works out best for you guys. Let's see. The website is here. It's the facultyweb.berry.edu/aceaerobic. It's going to have everything. It has all the PowerPoints for the entire semester. It has all the homework assignments ready to go. It has a few helpful links along the side, and it should pretty soon, I'll have practice exams up there. I'm just finishing working on those. I don't have a strict attendance policy. I would love to see you guys every single day. But if you don't make it, I'll be okay. Just make sure you get the material or watch it later on my fancy YouTube channel. Let's see. Same standard breakdown. We're going to have 3 exams, 15% each. A final exam that is cumulative but not standardized. So it's not an ACS exam. It's just a cumulative exam. That shouldn't be lab. Sorry, cross that out. You guys aren't taking lab. Should be, and there's a weird spacing in there. Anyway, move that over. That should be 15%, and this is going to be 20%. There we go. Sorry, typo. If you have in front of you, change that. So the homework, there's going to be homework every chapter, and so the homework's going to be weighted a little bit heavier because it is 10 to 15 problems about once a week. And this part just describes how I get to the grade, right? If you're between 93 and 100s, that's definitely an A. 90, 92, that's an A minus. Okay, we have 3 regular exams plus a final exam. Those are the dates. If you know in advance, because they are all Fridays, and I know we have some athletes in here, if you know you're going to be traveling on the Friday, try to work it out with me, if at all possible, to take the exam early. If you have to take it late, that is okay, but the exam will probably be different and more challenging just because you've had the extra weekend potentially to study for it. Final exam, December 10th, which seems super far away, but it's going to be here really fast. Definitely at the end of Monday class periods, we'll have a short 10-point quiz. Those will be definitely individual quizzes. We'll have a discussion about if you guys want to have group quizzes on Friday. I took it out of the syllabus this year, but if you guys feel like you'd like to have some extra quizzes in your groups, we can do that. Homework. It says at the end of each class period I will give a homework assignment, but I've already given you all the homework assignments on the very last page with the due dates. So you guys already have all the homework for the entire semester. So you guys overachievers, you can just go ahead and knock that out this week and then have the rest of the semester off doing homework. Grading homework, you are encouraged to work together on your homework problems. I don't have a problem with that if you guys want to work in groups on that. As long as everybody turns in their own individual homework set, so I have something to grade you personally on. There are going to be several seminars given throughout the semester. I require that you go to one seminar. It can be in chemistry, biology, animal science, physics, math, any of those areas. I don't care, but you just need to email me a kind of written paragraph that summarizes what you learned about. The chemistry ones, I'll have the dates for you on Wednesday. I think we'll have them locked in by then. And I should be able to get the biology ones and the physics ones pretty soon, because I think they're working out the last few details about when their seminar speakers are coming. The extra credit in here is if you attend more than one, you receive extra credit points put towards your quiz scores. So same as if you had me in any other class. If you do extra seminars, I drop the lowest quiz grade and I give you an automatic 10. So that's again. What else we got? You guys should be familiar with FERPA now. Don't have your parents call me. I don't want to talk to them. I'd rather just talk to you about if we're having an issue. If you do believe you need additional accommodations, I need that letter from Martha Van Sice as soon as possible so that we can get that worked out for you. And most importantly, academic integrity. On every single exam, I will have you sign a statement to the fact that you have not cheated and you have not witnessed anyone else cheating. The offenses, if I find to have a violation of academic policy, would be the first offense is you fail that quiz, exam, homework. Since homework is done in groups, very hard to cheat on the homework, unless I find you Xeroxing the answer book and just turning that in. That might be an issue. But no credit for that exam or quiz. Second offense you fail the course. So just so you guys are aware of that. I've had lots of people fail a particular exam or quiz because of academic integrity. I've never had to get to the point where I had to fail them from a course. So don't be trailblazers in that area. All right, here's our schedule for the semester. Today we're going to get started on chapter 1, which is intro, review of big topics, some thermo, some just in general. I'm sure how many guys have taken Bio 111? Bio 202? Animal Science 120? Okay. How many of you guys are familiar with pretty much all the building blocks of biochemistry? No one. Perfect. So apparently they're not covering anything this course is not to worry about, so we're good. We're good. All right, so there's our schedule for the semester, and then the last page of the syllabus is the homework. And you can see that your first homework assignment is due Friday. And there's 2 columns. There's the required problems. Those are the ones I want you to turn into me with your name stapled without the weird frilly stuff on the side. I want just plain sheets of paper, because otherwise they all get caught up in my other stuff and I lose them. Now for every chapter, I'll also give you some suggested problems. If they're suggested, it's code that's really required, but I'm not collecting them, which means they're fair game for quizzes and exams. Right? That's the fancy professor lingo. That suggested doesn't really mean suggested, it just means I'm not grading them. Hint. Big hint. Mm-hmm. All right, questions on the syllabus? Who's ready to get started? Just me. Oh, Sarah. Me and Sarah. Excellent. All right. I've never really used the .cam before, so it'll take me a second to figure out where to put all my stuff. I know, I asked Dr. Brett and Dr. Davis some tips on how to use the .cam. Mostly their tip was keep moving your hand so I'm not blocking all the words I'm writing.
You're fine.
All right, so chapter 1 is going to be a review of fascinating facts about biochemistry. And eventually I'll get a tip of where I should put all my sheets. There we go. Okay, so biochemistry introduction is the study of how life processes result from the interaction of lifeless molecules. So what that means, we're going to be concerned with really 5 big questions this semester. All right, so the first question is going to be, what are the chemical and 3-dimensional structures of biological molecules? So who had Dr. Davis for organic last year? Does everyone remember on the very first day of Dr. Davis's organic class, she talked to you about how structure determines everything? And she had you come up with an example where like 2 chemical compounds that look similar but act very differently, and then there's 2 like physical things that are similar in shape but act very differently. Like if you're dying in the desert of thirst, would you rather drink it through a regular straw or a coffee stirrer? Right? Those are both straws. One does a very specific job, like drinking water. One is just for stirring. That's pretty much the whole concept behind biochemistry. We have very few molecules to work with. They all have very specific things that they can do because of their structure. We're also going to look at how do biological molecules interact with each other. So a real easy example is going to be our enzyme and substrate. guys have talked somewhat about enzymes and substrates and how some enzymes can work with many substrates, can only work with one. So we're going to look at that in more depth. So another question is going to be, how do cells, put the E in the wrong spot. How do cells synthesize and degrade biological molecules? So how do we make peptides? How do we break down peptides? How do we make DNA? How do you break down DNA that's no longer useful? Big questions like that. Fourth question is going to be, what are the mechanisms for organizing biological molecules? So how do we, in a cell, which seems really tiny, but for a molecule is gigantic, like a swimming pool, in that swimming pool of information, how does DNA find the right next substrate? Or how does your peptide, as it's growing, find the right amino acid? Right? Because there's a million little pieces floating around there. How do you get the right part and the right string? And finally, the fifth question is, how is genetic material stored, transmitted, and expressed? So how do we get our genetic material to the right spot? How do we store it so it doesn't degrade? How do we then make more of it? And then how do we turn that genetic material into RNA, proteins, any other kind of material? Everybody excited about those 5 questions? Good, good. All right, can everyone read what I'm writing? Is it kind of sloppy? Do I need to zoom in? All right. So then let's talk about the chemical foundations of biochemistry. All right, apparently I need to pick a bigger font on my little sheets. That says real small. So it says chemical foundations, then it says living things are only made of a few elements. So as you know from organic chemistry, there's really only 4 main elements that make up all organic molecules. What are those 4? Carbon, oxygen, nitrogen, hydrogen. Excellent, those are the 4. So turns out that carbon makes up 62% of all biological molecules. Then we got a smattering of nitrogen at 11%, oxygen at 9.3%, hydrogen at 5.7%. And then we'll have a smattering of some other cations, right? Calcium, magnesium, potassium, sodium. So whatever is left over is going to be from that. So when you think about all the different things that are in your body, the fact that they're only really made up of 4 elements is fairly mind-blowing. All the different combinations you can make. And as a caveat to that, the chemical reactivity of I'm going to call them biomolecules, is limited relative to the reactivity of every single type of carbon compound you can make. Right, so there's nothing in your body that's explosive, even though you could easily make something explosive containing nitrogen, oxygen, a little bit of carbon. You could make gasoline containing just carbon and oxygen. There's currently, hopefully, unless someone had a crazy breakfast, no gasoline in your system right this second. So all these molecules that could potentially be very reactive are not so much, which means we're real limited in all the different things that your body can possibly produce, which is good. There's a good reason that we're going to be limited. So let's talk about how organic chemistry plays a part in this. So organic chemistry in the biological realm. Most molecules in a living cell belong to one of a very few functional group classes. You guys all remember some functional groups? If you don't, now is a good time to pull out those flashcards again and check them out again. And this is so important, I made it redundant. Chemical reactivity of biomolecules is limited. So, the type, this is in your textbook, so don't feel like you have to copy it down. So, the common functional groups that we're going to be concerned about and you're going to want to be able to recognize on sight: amines, NH2 or NH3s, alcohols, OHs, thiols, SHs, ethers, have an oxygen caught between two other R groups. We have aldehydes, which has a double bond oxygen next to a proton. Or we have ketones, which has a double bond oxygen next to two R groups. Carboxylic acids have this C double bond O next to an OH, or it could be deprotonated and just be O minus. We got esters, which is C double bond O, a no, that's not an H, so an R group. Amides, C double bond O next to an NH2. Imines, double bond N. And then these last group, which are the phosphorics. So either phosphoric acid, which is P double bond O, OH, or phosphoric ester, which is two acids connected to each other. So those are good. If you don't know those by sight, make a little star on that page and come back and just give yourself a quick little refresher course, because those are going to be, I'm going to say those are going to be the only functional groups that you need to know. So if that makes you feel better, that's how many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12. 12 functional groups. Those are going to be the only 12 I need you to know by sight. You probably already know them. It's just a little refresher. Check them out again. So let's talk about how those organic compounds fit into one of four major biomolecular groups. So these are going to be four groups that we're going to be most concerned about: amino acids, carbohydrates, nucleotides, lipids. And I'm going to ask that you're going to be able to identify each kind. So if I give you an unknown molecule, I want you to be able to tell me amino acid, carbohydrate, nucleotide, lipid. In each of these four categories, has a specific job in the body, which comes with specific chemical properties. I guess which is due to. All right, so we're going to take an in-depth look at each one of these four, which for most of you will be review. I'm sure you guys are all fairly familiar with these four categories. We're just going to take a look at each one of them, and I'll tell you some of the chemical properties or unique things about each one of these. All All right, amino acids. Why would I need amino acids? What do they go on to do? Proteins, right? They are our building. How many amino acids are there? 20 standard amino acids. If you don't remember your 20 standard amino acids, get those flashcards out also, because you'll need to be able to identify amino acids. I'll put this in a little star. For exam 1, and then by default, obviously, every exam after that, but definitely for exam 1. Able to identify 20 standard amino acids by structure, by name, and did you guys also learn the one letter and abbreviation for all of them? Now's a great time to go ahead and do that. Yeah, the one letter abbreviation for each one of them. I don't think abbreviation is MABs in it, but something like that. I'm going to apologize in advance. I'm a terrible speller, but for the most part, you can figure out what I'm trying to write. All right, so then let's talk about what makes an amino acid fascinating. Okay, amino acids get their really clever name because they have 2 functional groups in every one of them. Any guesses as to what 2 functional groups are in an amino acid? An amine, that's one of the 2. Carboxylic acid, an amino acid. So everyone has an amine group and a carboxylic acid. All right, those are our 2 functional groups we're going to be concerned about, our amine, an NH2, carboxylic acid, a COOH. Now, at pH 7.2, which we're going to assume is fairly standard for all living creatures, recognizing that there are some organisms that don't have a pH of 7, but we're going to pretend that all organisms live at pH 7.2. What happens to this amine group? I remember from working in chemistry. What is most, or any other class, what is most likely to happen to an amine? Will it be protonated or deprotonated? Let's start the easier one first. Let's do the carboxylic acid. Do carboxylic acids want to get one more proton or give one away? Give one away. So the carboxylic acid will really be in the form COO minus. Then let's go back and think about the amine. Is the amine going to give one away and be NH minus, or is it going to get one and become NH3 plus? NH3 plus, excellent choice. So in reality, when an amino acid is written, in most cases, you'll want to deprotonate one end and protonate the other end. Here's your fun word of the day. Use it at parties. People think you're really smart. This is actually a Zwitter ion. There's also a great Scrabble word, ZWITTERION. That would kill at Words with Friends if you get those combination of a letter in there. I haven't been able to make it work yet, but I keep trying. Okay, here we have our Zwitter ion, which means that it exists as both the deprotonated and protonated ends. So a Zwitter ion exists with both a deep, which means overall, what's its charge? Neutral. It comes out neutral. So even though it's charged, they cancel each other out and has an overall net charge of 0. So let me give you one example of amino acid. It happens to be my favorite one. So my one example, I guess I'll draw it up here in some blank space. Anyone off the top of their head can remember which one ends with a sh? That's all right. By the time I ask you Friday, or maybe next Monday, you guys will all be. This one's cysteine. Right, so we have our carboxylic acid group. We have our amine. What is the sh part of it? You can flip. Thiol. That's thiol. I love thiols, just in general. It's a fun fact about me. If I filled out a note card, favorite molecule, thiols. Not because they smell great, because thiols are ones that smell like rotten eggs, but because they really do amazing chemistry. So we'll talk some more about some thiols when we talk about hair and nails, because that is a lot of thiol chemistry. You ever wonder, has anyone ever had a perm? Few people. Well, we'll talk about why a perm works. And maybe I'll encourage everyone else to get a perm because of the fascinating chemistry, and the stuff that you get on your hair for a perm smells awful. That's because it's thiol. So anyway, we'll get some fun facts about hair and nails later. Questions about amino acids? All right, let's talk about some carbohydrates, which I recognize not everybody eats carbohydrates anymore. They are my favorite food group, just in general. So we'll spend a lot of time on carbohydrates just because they're tasty. So we're going to spend most of our time talking about simple carbohydrates, which are all the sugars: glucose, fructose, sucrose. Those are the ones that come to my head right now. The other one, which is spelled Fuctose, which I don't like to say because it could be easily misconstrued. But we'll also talk about that one because it's an interesting shaped sugar. I'll just always spell it out. So we'll spend most of our time talking about those guys: glucose, fructose, sucrose. Okay, we can draw them as a linear molecule. All right, so this happens to be glucose. And sometimes we'll draw them as a linear molecule so we can take a look at the enantiomeric carbon up here at the top and figure out if it's R or S. Man, that organic chemistry just keeps coming back, right? Enantiomeric, R or S. If it an R and S, we'll also use as D and L. You guys talk about how those are. Did you guys only use R and S? Use D and L? Excellent. I'll make a note. Go over D and L. So R and S is similar to D and L in terms of which-handedness each one of them are. But most likely in the body, this glucose molecule shows up as a cyclical molecule. So the same molecule will show up as a six-membered ring. And then once it becomes a ring, then we can talk about if it's in the boat conformation or if it's in the other conformations, depending on what it's going to go on to do. And then once in the ring, it can go on and polymerize and make a string of rings, which is how we get the cellulose. So cellulose is a complex carbohydrate that is a polymer of rings. By about 10 o'clock, I've missed my snack because I normally have a snack at 10 o'clock in the summer. And so a lot of my conversations will be food-based because I'm just waiting so I can have my snack at 11 o'clock. So when we talk a lot about food, this is why. Because I'm starving at this point. Plus, I taught BCC 100 last week, right? And every single BCC class, right, they give you cookies and candy, and that's all sitting up in my office still. I like a gazillion boxes of cookies. Megan's smiling because she brought half the candy to the class. And I still have all that. Then someone left their bag of candy when they came to visit me about scheduling. So then I have extra candy. So if anyone needs a pixie stick, I have some in my office. All right, let's talk about nucleotides. All right, what is a nucleotide used for? Could be two options. DNA and RNA, right? These are the building blocks of DNA and RNA. All nucleotides will contain three major parts. They're all going to contain a 5-carbon sugar. So the one we just looked at was a 6-carbon sugar because if you look back at its picture here, right, there is 1, 2, 3, 4, 5, 6. And when it rings up, the oxygen is in the ring and one of the ends is on the outside. So a 5-carbon sugar is going to look similar, and that's going to have 4 carbons in the ring and then some else stuff sticking off the edge. A nitrogen containing ring and a phosphate ester. So let me draw ATP, because ATP shows up a lot, not just in DNA and RNA, but it's one of our energy molecules. So adenine triphosphate. I'll start down here at the bottom, work my way up. So there is our 5-membered carbon sugar. Hanging out here at the bottom. Ribose. It's going to be stuck onto our nitrogen-containing ring above it. Right, one nitrogen-containing ring. That's our adenine. So what's the thing we're missing? We got a phosphate ester we got to stick off on the end. Then we got our phosphate ester hanging off the end. So the thing that's going to change between our nucleotides is going to be, it can either be this nitrogen-containing ring up here, that is different options, right? Uracil or whatnot. That's going to change. And then we could switch out our ribose for our deoxyribose, and then change this from, currently this is a structure that would go into RNA. We could change the deoxyribose, make it to a DNA option. So both of these, I guess I'll put stars over here. Star, star. Those are groups that can change. We could make, the phosphate ester will always be there. It's just going to depend on how many esters we put on there, right? ATP can break off and make ADP to make AMP. Vaguely familiar. I see a few people not willing to shake their heads, but I can see in their eyeballs. Yes. Yes, you guys have talked about ATP, ADP. Yes, excellent. Awesome. All right, let's do one last class of molecule, and then I'll let you go for today. Let's talk about lipids. Another set of molecules that I'm a big fan of. Because they are really, the most delicious food contains carbohydrates and lipids. So I do like talking about them. Okay, these guys don't have such strict rules as the first three examples. So our lipids is a much larger collection of molecules that can fall into this category. Let me draw an example molecule, and you'll tell me what one thing all lipids have in common. So let me draw one example. Does anyone happen to recognize what that one is? We may have not talked about it yet. This is cholesterol. Yes. Yep, it has mostly all saturated, happen to have one double bond, but mostly all saturated. Typically. Would this compound fall under the polar or non-polar umbrella? What do you think? Could depend on the location of the molecule. Looking at this one, it's just standing alone. If I had a bottle of cholesterol, which I do, up in my office, I use it for something else. But I have a big old jar of cholesterol. Would it be soluble in water or not? Not. If it's not soluble in water, polar or non-polar? Non-polar. So not water soluble. If it's not water soluble, but you as a human being are 90-ish% water, what function would all these lipids serve to do then? Where do you see them? In membranes. They're going to be used to keep your cells separated from each other. And so they're going to be kind of like gates that keep things in or out and keep all the cells separated. So we're going to see them in membranes and a few other fascinating places that we'll get to. All right, is everybody super excited about the semester? If that doesn't get you excited, just wait till Wednesday when we talk about thermodynamics. Okay, I will see you guys Wednesday.